Thank you very much for a kind introduction. I'm really excited to be here and to present my work. And I would really like to thank both Birnstiel Foundation and IMP for this amazing award. It's been a real honor for me. Thank you. So during my PhD, I was interested in understanding how mammalian cells can recognize and respond to different genetic invaders. And this is an important question because as much as 50% of our genome can be actually traced back to different mobile genetic elements, such as viruses and transposons, which invade our genome either from the outside or from within the cell. These elements have dual nature because, on the one hand, they pose a threat to the cell because their uncontrolled spread may interfere with genome function, for instance, through introducing mutations, disrupting genes, interfering with gene expression, or creating some harmful genomic rearrangements. On the other hand, however, they can greatly contribute to the evolution of our genome because they can provide the genome with a raw genetic material. So they can be a source of regulatory sequences, they can induce beneficial genomic rearrangements, modulate gene expression, and even create new genes. And in fact, in some cases, this can lead to really extreme phenotypic change. And to me, the most striking example is the single insertion of the allo element, which has been proposed to be a reason why we humans lost our tails. And you can see this absolutely crazy experiment from the Yanai's lab, which aims to model the consequences of such insertion in mice. And you can basically see that mice are born without the tail if they have this single allo element present within their genome. So I think it's clear that understanding the mechanisms by which cells cope with mobile genetic elements to, first of all, protect their genome so that they don't cause too much harm, but also to safely maintain these elements in the genome so that we can at some point benefit from these elements, is really of a fundamental importance. And my PhD project started by looking at how cells cope with one particular class of mobile genetic elements, which are called line 1 transposons. And these guys are the only autonomous mobile transposons in humans, and they occupy as much as 17% of our genome. And in fact, some of them can still jump from one genomic location to another using this copy and paste mechanism. What has been known before I started my PhD is that line 1 transposons are controlled by the epigenetic repressor complex called HASH, which is a complex of 3 proteins, TASOR, MPP8, peripherin, which has been discovered and characterized in the Lehner lab. And what HASH does is that it transcriptionally silences these line 1 transposons. So HASH basically sits at the genomic sites where line 1s are integrated and promotes the position of the classical silencing chromatin mark called H3K9 trimethylation. And by repressing this expression of line 1, basically HASH keeps these elements safe in check and prevents their jumping and spreading. So that was known, but the key unanswered question was how the HASH complex actually recognizes these elements when they first jump into the genome. How does it know this is a line 1 that it should silence, and how does it differentiate these elements from the rest of the genome? And to address this question, I decided to mimic this initial process of invasion by line 1s using simple fluorescent reporters. So typically such reporters would of course have a line 1 sequence inside, which consists of 2 open reading frames, ORF1 and ORF2, and would have a fluorescent reporter marker clone in between. And so now you can basically integrate that reporter into the cell and measure expression by flow cytometry. And what you can see here is that this reporter is silenced very strongly by HASH, because there is a massive difference in expression of the reporter in the cells, in the wild-type cells in gray, histogram, and cells that are depleted from HASH in purple. And here we're just knocking out HASH by knocking out one of the HASH complex members called TASOR. And this is just one example of this, but of course we tested many different ones. What you can see always is that these reporters are very strongly silenced by HASH, but you can also notice that these 2 histograms are not overlapping. So that really means that in every cell, every line 1 that randomly integrates into the genome is recognized and silenced by HASH, no matter where it integrates. So clearly there must be something about the line 1 intrinsic property of the line 1 that attracts HASH. And then I was able to quickly identify that this is this ORF2 fragment of the line 1 that is responsible for attracting HASH, because always the reporters that would have the ORF2 sequence were silenced, but not the one with the shorter fragment, ORF1. Then I really try hard to understand what it is about this ORF2 that attracts hash. And I did everything I could to really take it apart. I was testing small fragments of this sequence, deletion of these fragments, was trying to change the sequence. And actually, surprisingly, what it eventually turned out is that there's actually nothing special about the ORF2, because you can take a such reporter and replace this ORF2 sequence with almost any sequence you like, and you can see that almost all of them will be always silenced. And again, as you can see, comparing this shift between expression in cells that have hash and are lacking hash, so basically comparing the shift between gray and a purple histogram. Here I'm just showing you a few examples of such reporters that have basically different sequences cloned in place of ORF2. So this was really remarkable, because that basically means that hash can recognize and silence almost anything that invades our genome. And in fact, even if it sees it for the very first time, because I showed that sequences that are completely foreign to the human genome, so for instance, sequence of Cas9, which is frequently used for CRISPR-Cas gene editing, despite these cells never seen this sequence before, hash can still silence such reporter. And also, it doesn't really matter if these transgenes are integrated as a lentivirus or as a direct DNA sequence or a plasmid. They all end up being silenced by hash. But you may have noticed that different transgenes or different sequences are silenced to different extent. So what actually determines whether and how strongly these transgenes are silenced? And definitely length. They all needed to be relatively long to attract hash, and also adenine-rich. They also needed to have high adenine-rich content within the sequence. More importantly, they all needed to be transcribed, because when I took identical transgenes, one with the promoter, one without the promoter, I could see that only the one that has a promoter, so the one that is actually transcribed, attracts hash and gets this H3K9 silencing mark, as you can see here from these sequencing tracks from the chip experiments. And in fact, I also found why actually hash would prefer or would basically silence only transcribed elements. And this is because one of the hash complex members, called periphyllin, recognizes nascent, newly synthesized RNA. And I showed that this happened even before the H3K9 silencing mark is deposited, which really means that the RNA binding by hash is likely the very first event during the target recognition that actually then leads to this repressed silent state. And in fact, it's not that it's just any RNA that hash binds to, but hash associated with very specific subset of RNAs, which I showed by genome-wide analysis of hash-bound RNAs, and I will touch upon this later. Apart from these three features that I mentioned that are important for whether and how strongly transgenes are silenced, there was one more very important feature that these transgenes that were silenced by hash had all in common. And that was that they all lacked introns. And in fact, I really showed this is critical, because when I took these guys, which I know are normally silenced by hash, and inserted introns into them, then hash was no longer interested in silencing these. And you can see one example of such experiment, where you basically can see massive difference in expression for the reporter, intronless reporter, again, judging by the shift between expression in wild type and hash deficient cells, and the same reporter, but now with the intron, this difference is completely gone. And in fact, this also happens in the absence of intron splicing, because I showed that introns that are completely unspliced, they still have this protective effect. So these results were really remarkable, and they were really important because they suggested an unanticipated mechanism for how hash actually can recognize such a broad range of targets. And perhaps by using introns, hash could distinguish self-DNA, I mean, our own genes, because majority of our own genes have classical exon-intron structure, from non-self genetic invaders, because these guys usually have very compact genome and lack introns. I mean, that was a very attractive hypothesis, but of course the obvious question is, okay, what about DNA, self-DNA that is intronless? So what about our intronless genes, right? Because if this hypothesis was supposed to be true, then we should also expect that hash should silence our intronless genes, because they look like exactly invading DNAs because they lack introns, right? And very interesting background to address this, whether this prediction is right, is provided by the process of retrotransposition of cellular mRNA. Because this process generates intron-less genes. And it happens through the activity of line 1 transposons as well. And it happens when line 1 takes our cellular mRNA that comes from any random gene and then integrates it back into the genome at the distant location. And because the mRNA at this stage is spliced, that creates the intron-less gene copy of this initial gene somewhere at the distant location. So now we can take these pairs of genes, intron-less gene copy and intron-containing parent gene, which is really a great comparison because they are very similar in a sequence, but what is different is that one has introns and one doesn't. And we can check hash binding over these gene pairs. And what you can see here on this heat map, where I basically depicted hash binding over some of these retrotransposed genes, and basically where more blue means more hash binding, is that hash specifically associates with transcripts coming from intron-less gene copies, but simply ignores these intron-containing parent genes, which I think really nicely and elegantly makes the point that hash is specifically recognizing intron-less targets. That was really nice, but I also wanted to go further and probe whether this is true on a larger scale. So I decided to look at the hash-bound transcripts over basically genome-wide setup over 3 different gene groups. Intron-less pseudogenes, intron-less protein coding genes, and as a contrast, intron-containing protein coding genes. Each dot on this graph is a gene. The blue shaded area indicates genes whose transcripts are enriched for hash binding. And you can see immediately this asymmetric pattern of the volcano plot for both of these intron-less gene groups, with around 20% of them being significantly enriched for hash binding, versus symmetric distribution of the volcano plot seen for intron-containing genes, and only 5% of them being enriched for hash binding. Within these 5%, majority of these genes actually have very long exons, which we know also attract hash. So this data basically means that hash recognizes specifically intron-less transcripts coming from intron-less genomic glossi, but also illustrates that any deviation from the standard exon-intron structure can be also sensed by hash, for instance, long exons. And this intron-dependent recognition by hash has very important implication, because lack of introns is actually characteristic to retroelements, which are elements, genetic invaders, which undergo reverse transcription. And of course, within our genome, the flow of genetic information is from DNA to RNA, but these retroelements, because they are RNA derived, but they also want to integrate back into our DNA genome, they need to reverse transcribe. So they need to go from RNA to DNA during their life cycle. And intronless DNA is actually a natural product of this process, of this reverse transcription. So by recognizing intronless DNA, hash can actually control this unusual reverse flow of genetic information within our cell. So to summarize, the take-home message is that hash uses introns to differentiate itself from non-self DNA. And this ability really makes hash a novel component of the innate immune system. And likely also the very first epigenetic pathway that does not strictly rely on a memory of previous invasion in order to recognize the target. So in a sense, we can think about hash as an innate immune system that protects our genome against almost any invader, the same way that our immune system protects us against almost any pathogen. And these findings have not only very important implications for emergence of new genes and overall genome evolution, but also, of course, are highly relevant for almost any molecular biology experiment that involves transgenes, as well as potentially for gene therapies, and perhaps pharmacological inhibition of hash may prove useful to increase efficiencies of such therapies. Of course, none of this would be possible without continuous support from my PhD advisor, Paul Lehner. I'm really grateful for all the excitement for this project and all great discussions that we always had. I also want to acknowledge members of the Lehner Lab, in particular Stuart, who contributed to this work, as well as Sergio from AstraZeneca, who helped us with bioinformatic analysis, and Boehringer Ingelheim Funds for supporting me with PhD fellowship. And thank you for your attention.